And now we'll cover DNA mismatch repair. This is for fixes for mistakes of the polymerase that are not caught by the 3' to 5' exonuclease activity of the polymerase. As we discussed, there is that 3' to 5' exonuclease activity, and there is that reason the energetic favorability for 3' to 5' exonuclease activity versus 5' to 3'. And we covered that, I believe, in the DNA synthesis lecture. But just to remind you, these are very rare errors. The polymerase itself generally only makes mismatches about 1 nucleotide per 10 to the 5th nucleotides that it puts in, and then the 3' to 5' exonuclease catches or improves that rate by about 100-fold. And so now, for those of you who can't do the mental math, that means that we have errors present about every one nucleotide every 10 to the 7th nucleotides that are put down by the polymerase. The other thing that this DNA mismatch repair mechanism is for is fixing short insertions and deletions. So of course the most extensively studied mismatch repair system is in E. coli. And E. coli is special because it's E. coli. No, E. coli is special because it's got a mechanism that is only present in E. coli and a few other gram-negative bacteria. So this is very different from the gram-positive prokaryotes and the eukaryotic mismatch repair system, and we'll talk a little bit about that as well. But the difference here is primarily in the fact that the E. coli can tell the difference between a newly synthesized strand and a parental and template strand, and that's due to this methylation of the parental strands. The methylation is added specifically at GATC sites by deoxyadenosine methylase. So as the new strand is being synthesized, it will eventually be methylated, but this methylation activity follows the synthesis activity by about 2 minutes, which I think is funny. But it is about 2 minutes. And so we have a mismatch, though, in the strand that is not the parental strand, and the E. coli machinery can tell the difference between the parental strand and the new strand for those 2 minutes, because at the GATC site there is no methylation. So before the methylation machinery comes along, we have 3 major proteins that are involved in this mismatch repair, and they're MUT-S, MUT-L, and MUT-H. MUT-S, MUT-L, and MUT-H. We begin by MUT-S is a homodimer or a homotetramer, so it's either a dimer of MUT-S or a tetramer of MUT-S. That's going to bind to our mismatch. And so we've got 2 different versions shown here because there's going to be 2 different ways that this could happen. We could either have a cleavage of the newly synthesized strand on the 5' end of the damage, or we could have the cleavage of the newly synthesized strand at the 3' end. So that's why we've got 2 here. So I'll talk a little bit more about that in just a minute. But we have MUT-S is going to recognize the actual damage or the actual mismatch in the strand, and MUT-L will bind to MUT-S. MUT-L binds to MUT-S. This MUT-S, MUT-L complex will activate MUT-H, and here's where the difference is. So MUT-H will cut at the nearest unmethylated GATC site. So we've got a methylated GATC site in the parental strand, and we have an unmethylated GATC site in the newly synthesized strand. So MUT-H will either cut at the 5' side of the damage, or it'll cut at the 3' site of the damage. So MUT-H cuts where it is, if it's 3' or if it's 5', that's going to ultimately dictate which enzymes are going to be responsible for the exonuclease activity or the removal of the damage activity. So here we're looking at the 5' end cut, and here we're looking at the 3' end cut. And in both cases, we've got a helicase that's going to come in and start unwinding the DNA from the site of the cut. Here we've got it coming from the 3' end, and here we've got it coming from the 5' end. So the helicase single-stranded binding proteins will bind to the newly synthesized strand, and then of course we need ATP. So here you see the difference. When we got the incision on the 5' side, we're going to wind up having XO7 or RecJ doing this actual removal activity, versus if we have it on the 3' end, we're going to have XO1, XO7, or XO10 that are going to hydrolyze the next strand in the 3' to 5' direction. So this one is hydrolyzing in the 3' to the 5' direction, and on this side, when we got the 5' end, we're hydrolyzing in the 5' to 3'. So the result of this is that we're going to have the removal of the mismatch in a number of nucleotides. So it removes all of these, or it removes all of these. And remember, it's going to be going either from the 3' to the 5' direction, or it's going to be going from the 5' to the 3' direction. And so just like most of our other repair setups, we're going to have the DNA polymerase 3 helicase that's going to fill in from the 5' to the 3' direction. So here it's going to start here and go to the 3' direction, where the Nick will be ligated by DNA ligase. Similarly, when we have our Nick on the 3' side of the lesion, we're still going to have 5' to 3' activity, where ligase will close this, seal this Nick here. Then we have the GATC in the new strand methylated, and so everything's fixed. So the key for this is that MUT-H is the recognition for, or MUT-H will recognize this unmethylated GATC site. And MUT-H is what's different between the E. coli, other prokaryotes, and eukaryotes. So MUT-H is only present in E. coli and a few other gram-negative bacteria. And if we don't have MUT-H or these methylated GATC sites, we're going to require some other method to nick the DNA. And so here we'll transition now into looking at the eukaryotic system. So eukaryotes have homologs to the MUT-S and MUT-L proteins, but not MUT-H, as I mentioned. And so MUT-S and MUT-L in E. coli are going to recognize the mismatch and activate MUT-H to do the cleavage. But again, because the eukaryotes don't have MUT-H, MUT-L and MUT-S homologs are not going to activate a MUT-H homologs. So the 3 human homologs to MUT-S, which are going to be in complex to make MUT-S alpha, are called MSH2, MSH3, and MSH6. And I'm not interested in you remembering those names. I just want you to remember that this MUT-S alpha, which is part of the recognition complex for the actual mismatch, is not just a single protein. Similarly with the MUT-L alpha, it's going to be, these are 2 human homologs, MLH1 and PMS2, that are going to make the MUT-L alpha. So unlike in the eukaryotes where MUT-S and MUT-L are individual proteins, these are complexes of proteins that have to work together. So in human mismatch repair, we need a strand break on one side of the mismatch. And here I'm showing you the 5' directed mismatch repair. And figure 1223 in your book shows you both this 5' and the 3'. They're going to be very similar, a few different enzymes involved. If it's a 5' error or a 3', or sorry, 5' nick or 3' nick. And so a nick has to be present already on one side of the mismatch in order for this to happen. And so we think of this as happening a lot on the discontinuous strand, or like if we have an Okazaki fragment. So, but the key here is that MUT-S alpha and MUT-L alpha are going to recognize the damage. And then we get the PCNA, which is our clamp, and the RFC, which is our clamp loader. These together are going to induce MUT-L alpha to do the actual cutting. So we have to start out with a nick in the strand, and then we're going to nick the strands at these red arrows. So with ATP, of course. So XO1 is a nuclease that has 5' to 3' hydrolysis activity, and that's what's going to do the actual removal of this bit of DNA. And so it has to come in and remove from 5' to 3'. And so when you take a look at the 3' to 5', think about why it's important for our MUT-L alpha to cut on either side of our mismatch in order for that XO1 to do its activity. So once we have our nicked DNA, that's going to be removed from the 5' to 3' direction by XO1, we wind up having RPA, which is a single strand binding protein, that's going to bind to the remaining strand to stabilize this strand to be used as a template for DNA polymerase and ligase to fill it in and ligate the new strand, as we have done in the past. So, of course, the big question is, how do we know which is the parental strand and which is the newly synthesized strand? And I honestly don't know the answer to that for eukaryotes. But we can see that there are some similarities. It's, of course, a little bit more complicated in eukaryotes than prokaryotes, as is the general rule of thumb. But in the end, the same thing is accomplished in that we are going to excise our mismatch and then fill it with a polymerase and a ligase. Finally, we have the SOS response, and this is the SOS as in international distress signal. So there's DNA damage and there's a stress response or a response within the cell that leads to the fixing of these problems. And so this is sometimes when the DNA polymerases encounter abasic sites or cyclobutene pyrimidine dimers during replication that weren't already fixed. They get stuck there and they can't progress. And so it takes advantage of error-prone DNA polymerases. So normally our DNA polymerase 3 is pretty high fidelity, meaning that when it puts down the appropriate base in response to the template strand. But error-prone DNA polymerases are able to catalyze DNA synthesis across a lesion, like some mistake that DNA polymerase 3 wouldn't be able to work across for whatever reason. They work with these error-prone polymerases will work with very low fidelity, meaning that they don't necessarily pay attention to the template strand. And this type of synthesis is called translational DNA synthesis. Once the error-prone polymerase is done and takes care of the problem base, such as an abasic site or the cyclobutane pyrimidine dimer site, the normal polymerase can do its job and can come in and restart the normal DNA synthesis. The good news is that these error-prone polymerases in the SOS response do allow DNA synthesis to progress in cases of damage that haven't been repaired already. But of course, the big problem is that these are going to introduce mutations. And so it's kind of a good news, bad news situation. The SOS response itself is a multi-gene response to DNA damage in E. coli. So this is, of course, prokaryotic replication. And it's a multi-gene response, and we really mean multi-gene response. There are more than 40 genes that are regulated by LEX-A and REC-A in the SOS response in E. coli. LEX-A and REC-A are actually two proteins, and again, these are going to be regulating SOS response. So LEX-A is going to be bound to a regulatory site upstream of the operator or of the genes that are involved in the SOS response, including REC-A. This is, again, multiple genes, so we call it a regulatory site for operator for multiple genes. And when LEX-A is bound, we get no transcription. REC-A is eventually going to combine with single-stranded DNA to form a nucleoprotein filament that's going to have some activity. So when we have UV damage, for example, we get expression of REC-A. REC-A will combine with with single-stranded DNA. And this single-stranded DNA, normally we think of single-stranded DNA as being stabilized by single-stranded binding proteins. And in regular synthesis, when the polymerase is able to move along at the same rate as the helicase is unwinding our DNA, single-stranded proteins are able to bind the single-stranded DNA in front of the polymerase. But when we have damage due to UV or whatever chemical damage and the DNA polymerase stalls, the helicase keeps going. And so we're left with a longer than usual strand of single-stranded DNA. And this REC-A protein will bind to this single-stranded DNA in a 3' to 5' direction. When REC-A binds with the single-stranded protein, or single-stranded DNA, it activates and becomes a protease and cleaves LEX-A into inactive LEX-A. Inactive LEX-A can no longer bind to the operator, and then there's transcription. And specifically, we're looking at transcription of 3 different polymerases, the error-prone polymerases. We've got polymerase 2, polymerase 4, and polymerase 5, which is actually in a few different subunits. So again, when we have REC-A that is transcribed in response to UV, it's going to bind to single-stranded DNA. It's going to activate and have a protease activity that's going to inactivate LEX-A. When LEX-A is removed from the operator, from the SOS operator, we're going to have transcription of polymerase 2, polymerase 4, and polymerase 5, which are the error-prone polymerases. And the error-prone polymerases, so we've got DNA polymerase 2, polymerase 4, and polymerase 5. DNA polymerase 2 is usually used, it plays an important role because it restarts replication after a lesion has been bypassed. And generally speaking, it's pretty high fidelity, but it can synthesize over an abasic site or other potential mutation sites. It does introduce frame shift mutations, and so it is definitely error-prone. It's funny to think about something that's generally pretty high fidelity as error prone, but it is possible that it can go over the abasic sites and induce frame shift mutations. Polymerase 4 is generally very error-free or very error-prone, and the fidelity of that replication or synthesis is dependent upon the type of lesion. DNA polymerase 4 is interesting because it uses the same DNA 3 clamp assembly. So normally when we think about regular DNA synthesis, we think about DNA polymerase 3, the clamp assembly and the clamp loader to make the hollow enzyme really high processivity, or to increase the processivity of DNA polymerase 3. DNA polymerase 4 can interact with the same clamp assembly. And so what happens is that DNA polymerase 3 will be going along, and it'll hit an error, and it's unable to synthesize over the error or the lesion. And so DNA polymerase 4 comes in using the same clamp assembly, the same clamp loader, the same beta clamp, and it's going to do its job and put in a base there. Once that base has been placed, then DNA polymerase 3 is able to continue on, and so it takes over with the clamp assembly and continues moving along. The actual switching mechanism for that clamp assembly is totally unknown, but the key for DNA polymerase 4 is that it can synthesize across the cyclobutene thymine dimers, the 6,4 photoproducts, abasic sites, and large covalent lesion or covalent adducts. It not only fixes the lesion, but it can also introduce untargeted mutations in sites that appear to be undamaged, and so that still isn't really well understood as far as how it's going to be introducing new errors. And finally, we've got DNA polymerase 5, which is directly regulated by REC-A, as we showed before. It's not only that its transcription is regulated by REC-A, and LEX-A, as in DNA polymerase 4 and 2, but REC-A is going to play a role in the actual activation of the DNA polymerase 5 complex, which is called the mutasome. And the mutasome is a multi-part situation, or multi-part protein. And it can catalyze translational synthesis in several types of DNA lesions, including, again, just like DNA polymerase 4, it can do cyclobutene thymine dimers, 6,4 photoproducts, etc. So it's going to be doing some of the same things as polymerase 4. It's just going to act in a different way. It's it's very, very highly regulated because of its ability to introduce new errors. We don't want it to be going wild because we don't want to get a lot of new errors in the DNA as we go. So it's very tightly regulated. And it's basically not used, well, it's regulated in a way so that it can't be functioning when normal polymerases are functioning. So those are the error-prone polymerases, and that's what is going to be or these are the polymerases that are involved in this SOS response to damage that cannot be led or cannot be or has not already been fixed and cannot be synthesized by normal polymerase.